For decades, immunologists thought a specific molecule called CD23 had one very straightforward, honestly kind of boring job.
Right. It was totally typecast.
Exactly. It was classified as this simple receptor sitting on the surface of B cells, dealing mostly with mild allergies and immunoglobulin E. It was basically the allergy guy of the immune system.
The background character.
Yeah. But it turns out CD23 has been living a double life. We now know it is secretly the master survival switch for the most destructive, highly inflammatory cells in an asthmatic lung. So, welcome to today's deep dive.
Glad to be here. This is a fascinating one.
Our mission today is to unpack this really dense, cutting-edge paper published in the journal Cell Death and Disease in June 2026. It's titled, CD23 Sustains NKT17 Cell Homeostasis via IL-7-MTORC2 Signaling.
Which is quite a mouthful.
It really is. But okay, let's unpack this for you listening at home. Millions of people suffer from asthma and allergic airway diseases, and this paper completely dismantles how we view cellular survival in those respiratory pathologies.
It really does. I mean, we are moving away from just observing immune cells causing damage to actually identifying the exact metabolic lifelines that allow those cells to exist in the first place.
Right, like finding the power cord and just unplugging it.
Exactly. And the researchers didn't start this investigation by, just poking around in a petri dish or using a computer model. They started with actual human data.
Yeah, they were looking at patients experiencing severe asthma exacerbations. What were they looking at specifically?
Well, they utilized a single-cell RNA sequencing dataset. It's labeled GSE193816, and it was taken directly from human bronchial brushings.
Okay, single-cell RNA sequencing. Let's break that down for a second. That's vital here, right?
Oh, absolutely vital. Think about old-school sequencing like a smoothie. If you take a tissue sample and blend it up, you get an average of all the genetic activity, but that masks all the subtle changes of individual cells.
Right, there's fruit in the smoothie, but you don't know if the strawberry is doing something different than the banana.
Perfect analogy. Single-cell technology lets you analyze the genetic expression of every single individual cell in that airway separately.
So they're looking cell by cell at the bronchial mucosa of these allergic asthmatics. And one anomaly just jumped off the screen, right?
It did. They saw a massive spike in the expression of the FCER2 gene.
And for those keeping track, FCER2 is the gene that codes for this CD23 protein.
Exactly.
Now, seeing an allergy-related gene spike in an asthmatic airway might not seem revolutionary on its own. I mean, it's asthma, of course, allergy genes are up. But the plot twist comes from what was happening in the background.
Right, because simultaneously, the researchers observed heavily altered genetic signatures in a rare but exceptionally powerful type of immune cell.
And these are called invariant natural killer T cells, or iNKT cells.
Yes, and iNKT cells are fundamentally different from your conventional T cells.
How so?
Well, conventional T cells, they require protein fragments to be presented to them, and that process takes days, to prime the cell, let it multiply, and mount a full response.
It's slow, like calling in the regular army.
Right. But iNKT cells bypass that entirely. They don't look for proteins. They recognize lipid or fat antigens presented by a specific molecule called CD1D.
Oh, wow. So they react to fats, not proteins.
Yes. And because these lipid structures are so universal, iNKT cells function as a rapid response unit. Within minutes of activation, they just release massive amounts of cytokines to dictate the behavior of the rest of the immune system.
Cellular first responders?
Oh, wait.
If we're looking at human asthma data, there are different types of these responders, right? They aren't all the same.
Right. iNKT cells aren't a monolith at all. There are three main subsets, NKT1, NKT2, and NKT17.
NKT17, that's the one the researchers noted was dysregulated in the human data. Why is that specific subset so bad for asthma?
The NKT17 subset is pretty notorious in respiratory pathology. Its main job is to produce a cytokine called interleukin-17, or IL-17.
And IL-17 in the lung is bad news.
Devastating news. It drives what we call airway hyperresponsiveness,
which is that physical constriction of the airway, the thing that makes it terrifyingly difficult to breathe during an asthma attack.
Exactly. And over time, IL-17 causes lung tissue remodeling. It literally permanently thickens the airway walls. Plus, it acts as a powerful chemoattractant.
Meaning it calls in other bad actors.
Right. It pulls destructive neutrophils into the airways, just flooding the delicate lung tissue with even more inflammation.
Okay, so let's do a quick recap here to make sure we've got the foundation. Number one, we looked at human asthma data using incredibly precise single-cell sequencing. Number two, we saw the CD23 gene spiking. And number 3, at the exact same time, these hyperdestructive, rapid response NKT17 cells were going rogue and pumping out dangerous IL-17.
That's the exact setup, yeah.
But, in biology, just seeing two things happen at the same time during an asthma attack could just mean the massive inflammation is turning everything on at once. It doesn't prove CD23 is actually controlling those NKT17 cells.
Correlation is not causation, as we always say. So, to establish a real causal link, the researchers had to turn to a classic biological tool. They engineered mice to completely lack the CD23 protein.
A knockout mouse model. So they just deleted the allergy guy.
Yep. If CD23 is merely a byproduct of the inflammation, removing it shouldn't fundamentally change the T cells, right?
Right. So what happened?
When they analyzed the thymus, which is where T cells develop, and the spleen of these knockout mice, the overall number and frequency of iNKT cells dropped significantly.
Wow. So they just didn't develop. But here's where it gets really interesting for me. We need to know if all the different iNKT cells vanished evenly or if the loss was specific to one type.
And the precision here is what makes this finding so compelling. To figure this out, they used intracellular staining to look at transcription factor markers.
These are the markers that identify the specific subsets, right? Like name tags.
Exactly like name tags. So they tracked T-bet for NKT1 cells, GAT3 for NKT2 cells, and RR gamma T for the NKT17 cells.
RR gamma T.
Okay.
And what did they find in the mice missing CD23?
The NKT1 and NKT2 populations were completely unaffected. They developed and survived totally normally.
Wait, really? Only the NKT17 cells disappeared?
Only the NKT17 cells completely vanished.
That is wild. Think of it like a massive factory. You remove one specific floor manager, and suddenly the entire welding department vanishes without a trace. But meanwhile, the assembly line and the packaging departments just keep running, completely oblivious.
That's a great way to picture it. CD23 is uniquely required for this one specific asthma-driving subset to exist at all.
So how did they prove this actually matters clinically? I mean, a mouse missing some cells in its thymus is one thing, but does it stop asthma?
They tested exactly that. They challenged these CD23 knockout mice with a synthetic lipid antigen called alpha-galser.
Alpha-galser. And because it's a lipid, it specifically triggers those iNKT cells.
Right. It's basically used to artificially model a severe asthma attack in the lab. Now, in normal wild-type mice, this challenge causes severe airway resistance and massive inflammatory cell infiltration right into the lung tissue.
A full-blown asthma attack. But what about the knockout mice?
The CD23 knockout mice were virtually protected.
No way.
Yeah, their airway resistance was dramatically lower, and their lung tissue showed minimal inflammation compared to the normal mice.
Because without CD23, the NKT17 cells simply weren't there. And without those specific cells, the lipid trigger had no asthma pathology to activate. It was like pulling the firing pin out of a mechanism.
Exactly. We have a clear clinical outcome, but of course the mechanical mystery remains.
Right. Why are the NKT17 cells disappearing in the thymus? Are they just, failing to develop their identity, or are they forming and then literally dying before they can deploy to the lungs?
To catch the cells in the act, the researchers isolated whatever NKT17 cells they could find in the knockout mice and ran them through flow cytometry.
Flow cytometry. Basically sorting cells with lasers. What were they looking for?
They were looking for 3 highly specific molecular markers: annexin V, 7-AAD, and caspase-3.
Okay, let's slow down and explain those. Because they weren't just looking for dead cells. They were looking for the active mechanical process of a cell tearing itself apart from the inside, right?
Right, active apoptosis, programs cell death. So, annexin V binds to the cell surface during the very early stages of cell death. 7-AAD penetrates the cell when the membrane is fundamentally compromised.
This is actively leaking at that point.
Exactly. And caspase-3 is the critical enzyme. It acts as the cell's internal demolition crew. When caspase-3 activates, it systematically dismantles the cell's structural proteins.
And what did the flow cytometry show?
All 3 markers were elevated through the roof. The NKT17 cells in the CD23 knockout mice were actively undergoing apoptosis right there in the thymus.
They were self-destructing. But a cell only activates caspase-3, that demolition crew, when the failsafes keeping it in check collapse.
Yes. And the primary failsafe against that demolition crew is a protein called BCL2.
BCL2.
Right. It's an anti-apoptotic protein. Its entire biological purpose is to keep caspase-3 turned off and keep the cell alive.
So let me guess. When they measured BCL2 in these dying cells,
the levels had completely plummeted.
Wow. Okay, so let's do another quick recap of the mechanism here so we don't lose the forest for the trees. Number 1, without CD23, NKT17 cells vanish. Number 2, they vanish because their internal failsafe, BCL2, drops. And number 3, without BCL2, Caspase3 wakes up and structurally demolishes the cell.
That is the exact sequence. But it raises the next big mechanistic question. What caused the BCL2 to drop in the first place?
Right. If CD23 is the missing piece upstream, how on earth does it connect down to BCL2?
Well, they scanned the surface of the dying cells, and they found a massive reduction in a receptor called IL-7R alpha.
IL-7R alpha, so the receptor for interleukin-7.
Exactly, which is a known fundamental survival signal for T cells.
Okay, this is where we can map out a distinct division of labor within the cell, because classical immunology usually focuses on transcription factors, the job description, right?
Right, proteins like PLZF and ROR gamma T. They sit in the nucleus and dictate the cell's identity. They basically tell the cell, you are an NKT17 cell, your job is to produce IL-17.
But IL-7R alpha functions entirely differently. If the transcription factor is the job description, the IL-7 receptor is the paycheck.
Oh, I like that. Yes, the paycheck provides the metabolic energy and the BCL2 necessary to just stay alive.
And this paper proves a critical uncoupling between identity and survival. Because CD23 doesn't dictate the cell's genetic job description at all. It is part of a totally distinct upstream metabolic pathway.
Right, CD23 ensures the IL-7 receptor remains stable on the cell surface so the cell can actually receive its paycheck.
So no CD23 means the receptor drops off, the paycheck stops, BCL2 falls, and the cell executes itself via Caspase3.
Exactly, it's a beautiful chain of causality.
But rigorous science requires running the tape backward.
Right.
If the lack of CD23 causes the cells to die because they are starved of IL-7, we should be able to bypass CD23 entirely. Like just force feed them this survival signal and see if they come back to life.
And the researchers executed that exact rescue mission. They took the CD23 knockout mice and injected them directly with exogenous IL-7 for 3 consecutive days.
They manually provided the missing paycheck.
Manually handed them the cash.
But wait, if you just flood a biological system with a potent survival cytokine like IL-7, aren't you risking sending the entire immune system into hyperdrive? How do we know this rescue is specific to the NKT17 defect and not just a generalized inflammatory panic?
That's a crucial point. But the analysis showed it wasn't a generalized bloom across all T-cell compartments. The exogenous IL-7 specifically rescued the NKT17 compartment in the thymus.
Oh, fascinating.
Yeah, their overall numbers shot back up, and their capacity to produce that inflammatory IL-17 cytokine was completely restored.
So the specific defect caused by the missing CD23 was neutralized just by manually providing the IL-7.
Correct.
So going back to our factory analogy, the factory manager is removed, the welding department starts to collapse, but if you walk in and hand cash directly to the welders, they go right back to work producing IL-17.
Right back to work. But honestly, the most revealing detail of that whole rescue experiment was hidden in the transcription factors.
What do you mean?
Well, even with the cells completely rescued and pumping out IL-17, their core identity markers, PLZF and ROR gamma T, remained remarkably low.
Wait, really?
Yes. The IL-7 injection provided pure metabolic survival. It did not fix or reset their transcriptional identity.
That is a profound biological concept. The cell is functioning and producing its highly specific signature cytokine, even though the transcription factors that supposedly define its identity are suppressed.
It proves that for these specific effector cells, survival is function.
Survival is function.
Yeah. If you provide enough metabolic momentum through the IL-7 pathway, the cell will execute its baseline effector function regardless of what the core identity markers in the nucleus are doing.
Incredible. So they proved IL-7 rescues them. But did they pinpoint the exact biochemical engine that IL-7 was turning on to generate that survival momentum?
They did. It's the MTORC2 pathway.
Okay, MTOR. The mechanistic target of rapamycin, we've talked about this before, it is essentially the master metabolic sensor of the cell, constantly balancing energy production with cellular consumption.
Right. And it operates in 2 distinct complexes. This research specifically isolates complex 2, or MTORC2. When IL-7 binds to its receptor on the surface, it triggers a cascade that activates MTORC2 deep inside the cell.
And MTORC2 acts as a highly specific biochemical switch, right?
Yes. It phosphorylates another protein called AKT at a very specific amino acid residue, serine 473.
Okay, that's getting super granular.
Yeah.
So phosphorylation at serine 473, which we call PAKT SIR 473, is the ultimate molecular on switch for survival.
The measurements confirmed this entirely. In normal mice, PAC-AT at serine 473 was robust, driving the production of BCL2, but in the CD23 knockout mice, it plummeted.
And after the rescue injection?
After the exogenous IL-7 injections, PAC-AT at serine 473 roared back to life, halting the apoptosis.
Okay, I want to give our listeners a way to visualize this whole chain because there's a lot of acronyms. Let's try a mnemonic, like a visual memory trick.
Okay, let's hear it.
I'm picturing a CD player that's CD23 playing track number 7, which is the IL-7 receptor. That music turns on a motor, which is mTORC2. The motor acts AKT to ring a bell, which is BCL2.
A CD plays track 7 to run a motor that acts to ring a bell. I love that.
CD23 IL-7 mTORC2 AKT BCL2. If the bell rings, the cell lives. If the CD player breaks, the bell stops and caspase 3 destroys the cell.
That perfectly captures the entire signaling cascade in one image.
So if mTORC2 is the real hidden engine keeping these asthma-causing cells alive, we shouldn't need to manipulate CD23 or inject IL-7 at all, right?
Yeah.
Like if we want a new therapeutic approach for asthma, we should be able to just shut down that mTORC2 motor directly.
And that's exactly what the researchers did for their final proof. They left CD23 completely intact. They took wild-type mice and treated them with a highly specific mTORC2 inhibitor called JRAB2011.
They just drug blocked the motor. How long did they treat them?
For 6 to 8 weeks. And the results mirrored the CD23 knockout perfectly. The NKT17 cell populations drastically decreased, while their identity markers remained untouched.
So by directly blocking mTORC2, they severed the metabolic lifeline, and the asthma-causing cells just died off.
Yes.
This research takes us on such an incredible journey. I mean, from a broad data set of human lung tissue in asthma patients down to a single phosphorylated amino acid. We started with CD23, a molecule we assumed was just a straightforward allergy receptor on B cells. Now it is revealed as an upstream checkpoint in the thymus.
Right, maintaining the IL-7 receptor.
Which catches IL-7, which fires up mTORC2. mTORC2 flicks the switch on AKT at serine 473, which produces BCL2, which ultimately stops caspase 3 from dismantling the cell. This entire chain is what keeps these highly inflammatory asthma-driving cells alive in your airways.
And the therapeutic implications here fundamentally shift the paradigm of asthma treatment.
Because right now, severe asthma relies heavily on broad immunosuppressants.
Right.
Like corticosteroids, which carry massive systemic side effects, or bronchodilators that just temporarily force the smooth muscle to relax.
Exactly. We are just treating symptoms or using a sledgehammer on the immune system. But we now have a highly specific molecular axis to target.
Instead of blindly suppressing the immune system, we can design therapies that block the specific CD23 IL-7R alpha mTORC2 axis.
We can safely starve the exact NKT17 cells, causing the lung inflammation of their metabolic energy. We force them into apoptosis without altering their genetic identity, and crucially, without wiping out the rest of the patient's immune defenses.
You aren't changing who the cells are, you are just selectively turning off their life support.
Precisely.
Which leaves us with a truly fascinating concept to consider as we wrap up today. We now have concrete proof that we can manually starve highly destructive asthma cells by identifying and cutting off a backdoor metabolic pathway like mTORC2.
It opens up a whole new world of targeted therapies.
It really does. So I want to leave you, the listener, with this provocative thought. We've spent decades putting biological molecules into neat little boxes. CD23 is for allergies, PLZF is for identity. But nature doesn't care about our neat little boxes. If a common allergy receptor is secretly functioning as the survival switch for asthma, could we use this exact same metabolic starvation tactic against other incredibly complex diseases?
Oh, definitely.
What if severe autoimmune diseases, or even treatment-resistant cancers, are relying on similar hidden survival switches that we've completely mischaracterized in our textbooks, just waiting for us to map the right metabolic axis? Something to mull over until our next deep dive. Keep questioning the established science, and thanks for exploring with us.